Welcome to all of you who have joined us today for this webinar, The Importance of Physicochemical Properties for the Bioavailability of Pharmaceutical Compounds. My name is Rebecca Ruiz and I am the Principal Scientist at Pion. Pharmacokinetics are known as ADME. This is for absorption, distribution, metabolism, and excretion, and study how a drug is processed by a living organism. That means that it determines the bioavailability of a drug, and this is the effective concentration of the drug in the target tissue. When a drug is taken orally, the limiting process is absorption, and this means that the drug needs to be in solution, needs to be permeable, enough lipophilic, and the degree of ionization is going to help us to understand all these properties. Basically, if we know the physicochemical properties, we will know the pharmacokinetics of the drug and then its bioavailability. In other words, if we modify the physicochemical properties, we can promote the bioavailability of the drug. Let's talk about physicochemical properties. The drug in solution will be in these two forms, neutral or ionized, or a mixture of both. If the drug is mostly neutral, it will have a slow dissolution and won't be very soluble. But if it's ionized, it will have a faster dissolution and it will be more soluble. What determines when a drug is neutral or ionic is the pKa, the ionization constant, which defines at which pH there is a transition between the neutral and the ionized molecule, which will make a big difference in the way that it will behave in solution and so in the human body. For example, the neutral form is more lipophilic than the ionized form. That means that it can be absorbed and cross membranes by dissolving in the lipid phase of the membrane. However, ionic form doesn't cross membranes. The parameter which defines lipophilicity is the log P. So, pKa, log P, solubility, and dissolution can help us to understand absorption. Let's start with the ionization constant or pKa. I would like to explain the pKa property using the example of the thymol blue solutions. Thymol blue is an indicator, so changes color depending on the species in solution, meaning that each species has a different color. Let me introduce the distribution of a species graph with the pH. In the y-axis, we have the percentage of the species in solution, and in the x-axis, the pH. Thymol blue has two pKa's. The first pKa is 1.7 and the second 8.9, meaning that there is two changes in the structure depending on the pH. The first pKa is the pH where the 50% of the species in solution are neutral and monoanionic. The second pKa when the 50% of the species in solution are monoanionic and dianionic. If the solution is at low pH, below the first pKa, the sample is neutral and is red. If we increase the pH and we pass the first pKa, the molecule loses its proton. The main species is monoanionic and the color is yellow. Increasing the pH higher than its second pKa, 8.9, the sample loses the second proton. The main species is dianionic and the color is blue. Many drugs are ionizable molecules, then they have pKa's, so they have different species depending on the pH, and each species behaves differently. One of these forms is the one which will be the candidate for the target tissue. Once we know at which pH our sample is neutral, the log P will tell us how much lipophilic it is. So, if it's going to be able to cross membrane. Log P is the parameter which defines lipophilicity. P is the partition coefficient and it's the ratio between the neutral species concentrations dissolved into immiscible phases when the system is at equilibrium, usually water and octanol. In other words, the aqueous media simulates the bloodstream and the octanol is simulating the membranes. The higher the log P, the higher chance that the drug crossed the membrane, so it will be more lipophilic. The neutral species is the one which will be able to go through the membranes. However, the ionized species will not be absorbed effectively and they will stay in the bloodstream. Coming back to the case of the thymol blue, we can see that the neutral species, the one with the red color, is partitioning into the octanol layer, so it will be able to cross membranes, but is not going to be the case for the other two species, the monoanionic with color yellow and the dianionic in blue. They will stay in the water, so they will be dissolved in the bloodstream. I would like to introduce the BCS, the Biopharmaceutics Classification System. This classification was introduced in order to decrease the clinical trials and identify absorption problems after drug administration. The BCS classified the drugs into 4 categories based on 2 physicochemical properties, which are directly related to the absorption process, solubility and permeability. In class 1, we have molecules with high solubility and high permeability. These are molecules suitable for pharmaceutical purposes. In class 2, we have molecules with good permeability. Usually these molecules are big molecules with high molecular weights and high lipophilic, which makes the molecule less soluble. Then most of these compounds have their absorption limited by its solubility. Therefore, there is an interest of increasing the solubility of these drugs and promote them from class 2 to class 1. One of the strategies used to enhance the solubility of the drugs is the use of additives or excipients. So the drug can stay supersaturated in biological fluids for a period of time long enough to allow absorption. In the next slides, I would like to show you some cases where we have promoted drugs from class 2 to class 1, adding additives and using one of our instruments, Pion Series T3, with our patented method, the check-salt technique, to determine solubility. For the determination of the physicochemical properties, we have developed the Series T3, a fully automated potentiometer which determines pKa, log P, and solubility of the studied drug. The vials are 4 ml, and so the working volume is 1.5 ml of the media. The instrument includes an electrode to measure the pH, usually between 2 and 12, and temperature probe, and a set of capillaries to dispense the reactants and the media. A spectroscopy probe to collect the UV signal, allowing the instrument to use 2 techniques, the spectroscopic technique for UV active compounds and the potentiometric technique for the non-UV active compounds. A stirrer and a turbidity sensor to detect precipitation. The determinations are performed under argon or nitrogen atmosphere to prevent the absorption of the carbon dioxide into the solution. To understand solubility using our technique check-salt, I would like to introduce the graph concentration of the neutral species in solution with time. The sample selected for this example is benzofluomethiazide, an acidic molecule with 2 pKa's. At the beginning of the titration, there is not neutral species in solution as the sample is completely ionized, and so the concentration of neutral species is 0. Once the sample is completely dissolved, we want to force the sample to precipitate. For that, we are adding acid to change the pH to move it closer to the pKa and form neutral species. The more acid, the more neutral species, and when we pass the 2 pKa's, the concentration of the neutral species is too high that the solution crashes and precipitation started. At this point, we have the kinetic solubility, when the first precipitation is detected. After that, the drug is still precipitating, and the concentration of the neutral species decreases until it reaches the equilibrium. The intrinsic or thermodynamic solubility is determined. Before the sample precipitated, the solution was supersaturated, reaching a maximum concentration of the species in solution. However, This supersaturated solution is thermodynamically unstable and has the tendency to quickly return to the equilibrium state by precipitation to the most stable form of the drug, the crystalline form. In order to evaluate the supersaturation, we have two parameters: the extent of the supersaturation, the maximum concentration of the neutral species in solution before it precipitates, and the duration of the supersaturation, which is how long the supersaturated state stays in solution until the drug precipitates in its more stable form. The key here is to keep the compound in the supersaturated state when the drug has the maximum solubility longer enough to give the drug time to be absorbed in the system. One of the strategies is to use excipients or additives. And in the next slides, I'm going to show you some examples where we have enhanced solubility using plasdones as excipients. Plasdones are homopolymers of vinylpyrrolidone that can interact with the drug by hydrogen bonds through the nitrogen or the carbonyl group of the pyrrole ring. We have used different K types of these polymers, where K represents the degree of polymerization. Another plasdone used was S630, which is a copolymer of vinylpyrrolidone and vinyl acetate. This is the same graph as before. This is bendroflumesacetate without excipients. And this is bendroflumesacetate when plasdone K12 is added. When 50% of plasdone is added, the solubility increased and stays supersaturated for the entire duration of the experiment. I would like to emphasize that the supersaturation state is the solubility of the amorphous form, in this case promoted by the interaction between the drug and the excipient, and eventually will come back to a more stable form at the equilibrium with the most crystalline form. Other examples are shown in this slide: the acids diclofenac, ketoprofen, and warfarin, which behave in a similar way to bendroflumesacetate in the presence of plasdones, so long-time stabilization of the supersaturated state. We can observe that there is no difference in the behavior of the drugs when the different K types are used. However, we have observed that for some drugs, the increase of the supersaturation concentration is for a limited time. And after that time, they reach the solubility at the equilibrium, a more stable form, the crystalline form. In this slide, we have the acid isoxicam and the bases pindolol and olanzapine, which are affected by the different excipient types. So the duration of the supersaturation increases with the degree of the polymerization. Effect of the excipients on the pKa of the drugs. I would like to mention this study because when we add excipients, we can also modify the pKa. We have selected two types of excipients here: two cyclodestrins, carbazole and captisol, and one polymer, colidone, which is a plasdone. Cyclodestrins are microcyclic rings of glucose subunits, and the size of the cycle depends on the N, the number of the glucoside groups. For example, carbazole has 7 glucoside groups. More interestingly, they can adopt a toroidal formation like a vessel and creates an internal cavity highly lipophilic. This cavity can be modulated by modifying the R groups. For example, captisol has sulfonic groups which have mutual repulsion, making bigger the entrance of the cavity, and carbazole has the hydroxyl groups with less repulsion, making the cavity smaller than captisol. In this study, we have chosen two model drugs, an acid, diflunisal, and a base, trazodone. We can observe in this graph the pKa shift with the amount of excipient in solution. So the more the excipient is added, the more the pKa changes. For diflunisal, the increase of the concentration of the cyclodestrins carbazole and captisol shifts the pKa to a positive increase, to a higher pH, which confirms that the neutral species of diflunisal is interacting with the cyclodestrin, decreasing the concentration of the neutral species in solution and so disturbing the aqueous equilibria of the drug. As I previously mentioned, captisol has a bigger cavity and so we can observe a greater interaction, a higher effect of the pKa shift than carbazole. The interactions of diflunisal with colidone are different in comparison with the cyclodestrins. The pKa shift is smaller and negative. This behavior could be explained due to the fact that the anionic species of the drug is the one that is interacting with the excipient. Opposite behavior is observed with trazodone, a base. In the case of the cyclodestrins, the increase of the pKa shift with the increase of the amount of the excipients is to a lower pH, which confirms that the neutral species is interacting with the excipient and so the concentration of the neutral species in solution decrease, disturbing the aqueous equilibrium of the drug, causing the pKa shift to a lower pH. It is observed that carbazole has a greater interaction than captisol, which could be due to the electrostatic interaction between the negative groups of captisol and the protonated species of trazodone. In the presence of colidone is observed a positive increase of the pKa shift. The interaction of trazodone with this excipient is different in comparison with the cyclodestrins, which could be explained by the interaction between the protonated species of trazodone with the excipient. So the pKa shift depends on the nature of the excipient, the interaction drug-excipient with the ionic or with the neutral species of the drug, and if the drug is an acid or a base, because the ionic species of the acids are anionic, negative charge, and for the bases are cationic, positive charge. We are going to talk now about dissolution. One of the strategies to enhance dissolution is the use of co-crystals. This slide shows the aqueous dissolution study of adefovir and its co-crystals, with the coformers resorcinol and orcinol. For this example, 15 ml of media was used, and so any of the Pion fiber optics instruments can perform these experiments. For this study, individual dissolution experiments of 30 minutes each were performed in aqueous media at the biorelevant pHs of 1.5, 4, 5.5, and 7.4, as these are the pH values at the different regions of the GI tract. This study is published and you can find the reference in this slide. In this slide, we can observe the dissolution plots. The dissolution of adefovir, the red plot, decreases as the pH increases. The reason for this is the ionization state of adefovir. Adefovir is a base with a pKa of 3.8, and so at pH 1.5, adefovir is positive charge, which helps dissolution. However, at pHs values higher than its pKa, at 4, 5.5, and 7.4, the percentage of the neutral species is higher and the dissolution is slower. The same behavior is observed with the dissolution of the co-crystals, the yellow and green plots. The dissolution profiles obtained at pH 1.5 show an initial rapid dissolution for the co-crystals adefovir resorcinol and adefovir orcinol between 1 and 3 minutes, followed by a plateau where the solution becomes saturated and the amount of API dissolved stays constant until the end of the assay. We can see that all 4 pHs show the same behavior. The co-crystals dissolve much faster at the beginning of the experiment, and the percentage of the drug dissolved at the end of the assay is higher for both co-crystals in comparison with adefovir alone. The experiment was repeated including a third co-crystal with the coformer hydroquinone and in biphasic conditions. In this case, the dissolution consists in 4 pH sector experiment of 30 minutes each in a starting aqueous volume of 14 milliliters, where decanol is added in the second sector at pH 4 to simulate the contact with the lipid membranes. Then the experiment started at the gastric conditions at pH 1.5. The pH changed to 4 in the second sector, and it's after the adjustment of this pH when the lipid layer decanol is added. At the end of this sector, the pH changed again to 5.5 and then 7.4. 2 UV probes are used for the quantification of the drug in the aqueous and the lipid layer, and also both layers are stirred at the same time. In this slide, we can observe the biphasic dissolution plots, adefovir and the 3 co-crystals, hydroquinone, orcinol and resorcinol, through the 4 pHs and the addition of decanol in the second pH sector. We can observe that the dissolution of hydroquinone co-crystal dissolves slower at pH 1.5 in comparison with the other co-crystals, with resorcinol and orcinol as coformers. So this co-crystal does not improve the dissolution of adefovir as the other two. The main differences appear once the decanol phase is added. Just after adjusting the pH to 4, the addition of the lipid layer mimics the flow of the compound from the stomach to the intestine, and the concentration of adefovir in the aqueous phase, the red plot, decreases because the adefovir is partitioning into the organic phase, the purple plot. More partition is observed when the pH is higher than the pKa of adefovir, which is 3.8, where adefovir exists in its neutral form. Then the maximum concentration of adefovir in the organic solution is reached at pH 7.4 in all the experiments performed. We can observe that adefovir permeates to the organic layer faster with the co-crystals resorcinol and orcinol than hydroquinone. Then resorcinol and orcinol co-crystals are better candidates as they facilitate adefovir permeation through the biological membranes. I would like to introduce the Pion SDI2, a surface dissolution imaging instrument. The SDI2 provides high-resolution images of the samples as they dissolve. It's capable of capturing images at 2 wavelengths during the same assay, UV and visible. The UV data is used to calculate sample concentrations, so the drug release, and the visible light can be used to follow the physical changes that the sample experiences as it dissolves. 2 flow cells are used, the compact flow cell and the whole dosage flow cell. The compact flow cell is designed for powder samples. The whole dosage flow cell is based on a USB Type 4 flow cell design and is used for the dissolution of larger samples, like tablets or capsules. Basically, the absorbance values determined for each pixel are assigned a color, and this color is based on their intensity to build up a color image of the flow cell and the dissolution which is taking place. The dual wavelength analysis allows for both the generation of the concentration versus time dissolution profiles, as well as quantitation of the physical changes to the tablet, such as swelling or disintegration. In this study, I would like to show the behavior of 3 different coated tablets when the media changes from gastric at pH 2 to the intestinal conditions at pH 5. So depending on how the tablet is coated, the API can be released in the target organ. For this example, the whole dosage flow cell was used and the tablets were located inside the cell as is shown in the slide. On the left-hand side, we see the instant release formulation. We can see that the tablet breaks immediately just at the start of the dissolution experiment at pH 2 in stomach conditions, exposing the active ingredient to the dissolution media. We see some dissolution and disintegration during the gastric phase, followed by a large release of drug as the media is changed to the intestinal environment. In the center, we have the enteric coated tablet. Which displays no changes during the gastric phase. After the change of the media to intestinal conditions, we have a short lag as the coating of the tablet is dissolved, followed by rapid and complete breakdown of the tablet and a vast release of the drug inside. On the right-hand side, we have the extended release tablet, which shows a much less dramatic change compared to the other 2 examples. Upon the media change, the tablet slowly starts to hydrate and swell as it takes on water. This allows the drug in the tablet to gradually diffuse to the surface of the tablet and to dissolve into the media. We don't see any bursting or disintegration of the tablet, and the drug release is reasonably consistent through the duration of the dissolution assay. This case shows an experiment using now the compact flow cell to study the enhancement of dissolution of dipyridamole using HPMC acetate succinate in different ratios as an amorphous solid dispersion. The dissolution of the samples was monitored using UV light. The images on the right-hand side show snapshots of the dissolution videos at 10 minutes. These color images clearly show the dissolution enhancement of the excipient, which is mirrored in the dissolution curves captured from the same experiment. Then, increasing the proportion of HPMC acetate succinate in the dispersion increases the dissolution rate of dipyridamole. This is my last case study. I'm going to talk about polymorphism. This is the same graph that we have seen before. So we have here the neutral species concentration in solution with time. However, we don't have an excipient here. This is just the behavior of a drug on its own. In this example, we can see how solubility changes due to different forms. The first precipitation is an amorphous form with a higher solubility, which is temporarily stabilized. However, after 20 minutes, a change in the form of precipitation is observed from amorphous to the more crystalline form, which is more stable and has a lower solubility. There are compounds that precipitate in different forms, meaning that each form has different solubility, and using our method CHECKSOL, we can determine how many forms the same drug has and determine their solubility. In this case, we need to add an excipient to prolong the supersaturation state of the first form and prolong longer enough to give enough time to the drug to be absorbed in the system. However, I wanted to show you this example as we verified that actually we had two precipitations with two different forms of the same drug. We collected the first and the second precipitation. This slide shows the two precipitates collected, the first precipitate and the second precipitate. The precipitates were centrifuged for 2 minutes, and if you carefully observe, we have here what appears to be an oil drop, which is coming from the first precipitate, the amorphous form, and a particle which is coming from the second precipitate, the more crystalline form. We collected the oil and observed using the polarized microscope. The first screenshot shows small oil drops, and with time we can observe how they started to convert to the more crystalline form. We also observed the second precipitate through the polarized microscope, confirming that it was the second form, the more crystalline form. And we have just arrived to the last slide, the concluding remarks. So, during preclinical drug development, the number of compounds decreases from hundreds or thousands to the one which enters the clinical trials. To understand the reasons for candidate failure, there has been considerable interest in understanding and optimizing the physicochemical properties such as ionization, lipophilicity, solubility, dissolution, and permeability as the key to developing a successful pharmaceutical active ingredient into a drug. Pion Series T3 determines PKA, Log P, and solubility using our method CHECKSOL, parameters which are recognized as setting the industry standard. Studies with excipients can help to predict which excipients will be more effective in extending and/or prolonging supersaturation and improving dissolution. The use of additives can modify the ionization constant, the PKA, of the drugs, promoting their bioavailability. The use of coformers to obtain co-crystals can improve dissolution of the API. Coated tablets as instant release, enteric coated, extended release can release the API in the target organ. The intrinsic solubility of different forms, amorphous, crystalline, could be determined using the CHECKSOL method in order to select which form could be promoted in solubility. Thank you for your time, and please let me know if you have any questions.